Hello everyone, my name is Cécile Herat and I am a biology researcher in the Cytogenetics Laboratory of the French Alternative Energy and Atomic Energy Commission. In this lab, we are experts in biodosimetry and we develop different cytogenetic methods to retrospectively estimate the received dose by individuals. We are also using these techniques to answer mechanistic questions by analyzing chromosome rearrangement. In this tutorial, I will particularly explain you the multifish or M-fish staining method that allows building the karyotype by differentiating each of the 23 or 24 chromosomes. I will tell you more very soon. After the chromosome preparation by the different methods and the spreading on slides, DNA has to be stained for further acquisition using a fluorescent microscope in order to allow for the different chromosome observation. The M-fish staining consists in labeling DNA using probes specifically recognizing each chromosome sequence. Therefore, it allows for the visualization of interchromosomic rearrangements such as translocation and unstable aberration like dysentery chromosome. In this MOOC, I will describe in more details the M-fish protocol using the commercial probes from Metasystems. This staining can be performed on chromosomes obtained after the classic protocol of metaphase for DCA assay, but also on premature chromosome obtained with a PCC assay. Here is an example. On the left picture, you can see the M-fish staining of metaphasic chromosome and, on the right, the resulting karyotype. You can observe 2 dysentery chromosomes and 1 translocation. The latter will not be detectable with the conventional staining. Different cytogenetic methods are used in biodosimetry and allow the determination of the received dose by individuals. The dysentery chromosome assay, or DCA, allows, with the use of Geza staining, the scoring of dysentery chromosomes and rings, while the telomere and centromere staining does not modify the main principle of the technique, but facilitates the analysis and improves the detection of chromosome aberration. The premature chromosome condensation assay, or PCC, allows for scoring of chromosome fragments and the determination of excess fragments. In this case, the fish staining results in specific scoring of dysentery chromosome, rings, and acentric fragments. So, the analysis is a little bit longer, but more precise and informative. The cytokinesis block micronucleus assay, or CBMN, allows for scoring of micronuclei after blocking the cell cycle in the cytokinesis step. The 3 techniques are explained in details in 3 other specific tutorials that are also available in this YouTube channel. In this slide, you can see the 3 types of staining: Giemsa, telomere and centromere, and M-fish, for both DCA and PCC assay. Try to spot the chromosome aberration. Good luck! Now, we will see the precise protocol of the M-fish staining using Metasystems probes. I will try to provide you with the maximum of details and the tricks that we have developed. First, you can see the list of materials you need for the staining, the necessary laboratory equipment and the various reagents. Pay attention to work under the chemical wood when indicated, since some solutions are toxic. Here is the list and the composition of the different solutions that you need to prepare. The day before the staining, you can already thaw the slides or take them out from the refrigerator. I remind you that cytogenetic slides can be kept for years at -20 degrees. Usually, the slides that are stained using M-fish have previously been observed with other staining methods and therefore will need to be destained through watching. After cover slip removal by a regular PBS prolonged incubation, we incubate 30 minutes in PBS twin 0.5% at 37 degrees. Before starting, you can prepare the solution as indicated in the slides. The first step consists of preparing the slides by fixing the chromosomes in formaldehyde. This step needs to be performed even if the chromosome had already been fixed previously for performing another staining. Don't forget to work under the chemical hood. We are using porcelain boxes for the different incubations. After washing, the slides have been rinsed in PBS. Now I am showing you the step of fixation. I transfer the slides from PBS in formaldehyde 4%. I will incubate them for exactly 2 minutes. After rinsing in PBS, the slides are transferred in 0.1 ECC at room temperature. The next step is the DNA denaturation. We first transfer the slides from 0.1 SSC to 2x SSC solution preheated at 70 degrees in a water bath. We incubate for 30 minutes. Then, we move the 2x SSC box to a 37 water bath and let it cool down for another 30 minutes in order to allow for the renaturation of repeated sequences. We rinse for 1 minute in 0.1 SSC and continue the denaturation. So, we incubate for 1 minute in sodium hydroxide 0.07 N. Then, we continue with 2 successive rinses, first in 0.1 SSC and then in 2x SSC, both at 4 degrees. Finally, we proceed to drying and incubating in ethanol baths of increasing concentrations. Ethanol has a property of triggering dehydration. Here we transfer successively the slides from ethanol 30% to ethanol 50%, then 70% and finally 100%. Each incubation lasts 1 minute. Finally, we remove the slides and place them in a specific rack. The second thing to do is to prepare the M-fish props. You have to calculate 15 microliters per slide. The denaturation consists in heating the prop solution for 5 minutes at 75 degrees. After 5 minutes, we stop the denaturation by placing the tube in ice for a few seconds. Then we transfer in the 37-degree water bath for 30 minutes to provoke the pre-hybridization. Now that both the slide and the probe are ready, we can start the hybridization process. Before starting, try to engrave on the back of the slides the chromosome spreading area in order to optimize as much as possible the deposit of the probe solution. Lay your slide down and take 15 microliters of the probe solution. Distribute in tiny drops on the slide as I am showing you. Then, protect with a plastic cover slip that you have previously sealed from a freezer film for the delimited area. Try to be meticulous when you drop the cover slip to avoid bubbles. If you get some, please try to remove them. The hybridization will then occur in a box containing 2xSSC with 50% formamide as indicated previously in the protocol. Here we use a homemade hybridization box. It consists in a plastic box with a central platform. On the side gutter, we have placed paper strings that we will generously humidify with the hybridization solution. Since we will incubate the slide for a long time, it is necessary to tape the box or find any other way to seal it in order to prevent the hybridization solution from drying out. For this staining, the incubation is quite long and will take place in a 37-degree incubator. A bacteria incubator set at 37 degrees could also do the job. You just need to find a way to maintain the temperature. We will incubate the samples for 4 to 5 days in the dark. 4 days is enough, but if you want to be sure to have a strong signal, you can wait 5 days. 4 or 5 days later, we will proceed to the slide washing. Refer to the solution list for the composition of the two washing solutions. We unseal the hybridization box, and since it contains formamide, we do it under the chemical hood. We remove each cover slip using a pair of tweezers and watch first in 0.4x SSC for 3 minutes at 72 degrees. Then, we perform a second washing in 2x SSC containing twin at 0.05% for 3 minutes at room temperature. After that, we rinse in PBS. The DAPI staining that follows consists in simply incubating the samples for 5 minutes in the DAPI solution, prepared at a concentration of 100 mg per milliliter, before washing in PBS for another 5 minutes. The DAPI solution can be kept up to 1 week at 4 degrees. When the slides are ready for mounting, we take them out and dry the back. We take p-phenylenediamine or PPD with a Pasteur pipette. We drop PPD on the slide and put the cover slip from the bottom to the top. In order to remove the excess of PPD and destroy untrappable bubble, we put the slide between paper towel and press gently. The slides are now ready for observation and acquisition with a fluorescent microscope. After the DAPI staining, the slides are ready to be analyzed. In the meantime, they can be stored either at 4 degrees for a few weeks or at -20 degrees for years in specific plastic storage boxes. For the microscope acquisition, you need to have access to a fluorescent microscope with DAPI channel and the 5 following filters: aqua, green, orange, red, and near-infrared. In our lab, we use the Metafair acquisition software, and I will show you the main steps for image acquisition using this software. First, if you didn't do it previously for the analysis of other staining, you have to create a gallery with a 10x objective. The software will automatically capture the cell of interest. You can make either an automatic reader of metaphases or PCC. After cleaning the slide with ethanol, you can start with either the establishment of the gallery or the EMFISH staining acquisition. The gallery is established with a 10x magnification. We program the microscope to acquire metaphases, and then it captures all of them in the predefined area. After the triage of the gallery, we start the final acquisition with the 6 fluorescent channels. The microscope will acquire successively the different channels to obtain the final image that allows for the definition of the karyotype of the cell.
The EMPHI staining seems to be very informative, and finally it is the only staining that allows detection of stable aberrations such as translocations, but it also takes so much time. When are you really using it? Because I guess that you cannot apply it in case of emergency for direct dose assessment.
You are right, this assay is mainly used for retrospective dose assessment when dicentric chromosome detection is no more accurate. Indeed, the rate of dicentric ring and acentric fragment decrease along each cell division. About 50% of dicentric chromosomes are transmitted to the daughter cell. It means that after some months, the dose determined with classical technique is inferior to the initial dose. Even if some algorithm allows the modelization of the decrease, the better method is to quantify stable aberrations that remain stable during decades. Moreover, this technique is also interesting for fundamental research to characterize the phenomenon of DNA reparation and recombination.
You say that I can apply the EMPHI staining on my old slides. Is there really any problem?
Yes, slides can be kept at -20 degrees for years without any damage. Just close them the day before the staining. In case of previous staining, you just have to remove it. However, when you are making a Giemsa staining, it is better to remove it rapidly in the following days. Another thing to note, multi-fish staining is an expensive assay but also very informative. It is usually the last staining performed on slide. After this staining, following one are no more as reliable.
Just a short comeback to the protocol. It seems like a simple recipe. Different incubations for fixation, protein digestion, denaturation, and hybridization. But what seems more obscure to me is the image acquisition and the analysis. Is it actually so evident?
You are right. It's not a complicated assay. You just need to follow step by step the protocol and be vigilant to respect the time and temperature of each incubation. Regarding the microscope capture, the way to do differ from the different instruments and software. That's why I didn't describe deeply this step. Anyway, the principle remains the same. If you didn't make it before, you need to make a gallery, using the 10x magnification and the channel for the DAPI acquisition. Either you have in your software an automatic capture of metaphases and PCC, or you can make it by yourself in saving the coordinate of each cell of interest. In case of automatic detection, you need to make the triage of the gallery. At that point, you are ready for the final capture using the 5 filters already described with the 63x objective. I cannot tell you more about time of integration. It really depends on each microscope. After export of the images, you can analyze them with a dedicated software. We are using ISIS, but for sure there are some others that will create automatically the karyotype.